Caspofungin beta-D-glucan synthase and thereby disturbing the integrity of the fungal cell wall. Caspofungin was the first inhibitor of fungal beta-D-glucan synthesis to be approved by the United States Food and Drug Administration. Caspofungin is administered intravenously. Caspofungin has been effective in treating fungal infections caused by Aspergillus and Candida species. It is a member of the echinocandin family, a new class of antifungal agents with broad spectrum of activity against all Candida species. In comparison to treatment with either fluconazole or amphotericin B, all three drugs in this class have been demonstrated to be highly effective or superior in well-defined clinical settings including invasive Candida infections, Candida esophagitis and candidemia. Higher minimum inhibitory concentration of these agents has been observed against C. paracillosis and C. guillemodi. The following summarizes Mike's susceptibility for a few medically significant organisms. Caspofungin acetate for injection was originally approved by both the Food and Drug Administration in the US and the EMA in Europe in 2001. Its currently approved therapeutic indications by both organizations include the empirical therapy of presumed fungal infections in febrile, neutropenic adult patients and for salvage therapy in patients' treatment of invasive aspergillosis in adult patients whose disease is refractory to or who are intolerant of other antifungal agents. Additionally, the FDA approval includes indication for the treatment of candidemia and some specific Candida infections, and the EMA approval includes indication for the treatment of general invasive candidiasis in adult patients, slowly metabolized by peptide hydrolysis and N-acetylation in liver. Therefore, in case of liver impairment, the dose needs to be reduced. Caspofungin also undergoes spontaneous chemical degradation to an open-ring peptide compound, L747969. Additional metabolism involves hydrolysis into constitutive amino acids and their derivatives, including dihydroxyhomotyrosine and N-acetyl dihydroxyhomotyrosine. About 36% of patients refractory to other therapies responded well to caspofungin therapy, while even 70% of patients intolerant to other therapies were classified as responders. Direct comparative studies to other drugs in the treatment of invasive aspergillosis have so far not been undertaken. Known hypersensitivity to caspofungin acetate or any other ingredient contained in the formulation contraindicate its use. The concomitant use of caspofungin and cyclosporine in healthy volunteers led to a more frequent increase of liver enzymes than noted with cyclosporine alone. Combination treatment is only indicated if the potential benefit for the patient outweighs the potential risk. Dosage reduction in patients with moderately impaired liver function is recommended. No clinical data exist regarding the use of caspofungin in patients with severely impaired liver function. Reactions due to histamine release have been seen. Healthcare providers should carefully watch for these reactions. In a few patients with infections caused by Candida albicans, mutants with reduced sensitivity to caspofungin have been noticed. Currently, there are no data regarding development of resistance in other fungi than C. albicans. Caspofungin has been shown in animal studies to have embryotoxic properties, and therefore has been assigned to class C. It should only be given to pregnant women if the benefit to the mother clearly outweighs the potential risk to her fetus. The drug is found in the milk of lactating rats, but it is not known whether this is seen in humans. Thus, lactating women should be treated cautiously. Ordinarily, no dose adjustments are necessary. However, greater sensitivity of some older individuals cannot be ruled out. Caspofungin is FDA approved for pediatric patients 3 months and older. Dosing is based on body surface area as calculated by the Mosteller formula. Compared to amphotericin B, caspofungin seems to have a relatively low incidence of side effects. In clinical studies and post-marketing reports, the side effects seen in 1% or more of the patients were as follows. Additionally, infrequent cases of symptomatic liver damage, peripheral edema and swelling, and hypercalcemia have been seen. One case of anaphylaxis has also been noted. Resistance in C. albicans has been described. but is currently still rare. The mechanism is probably a point mutation in the beta-D-glucan synthase gene. The mean duration of therapy in previous studies was 34 days. Some patients were even healed by a one-day treatment. However, a few patients were treated for as long as 162 days and tolerated the drug well, indicating that long-time use may be indicated and tolerated favorably in complicated cases of aspergillosis. Generally, the duration of treatment is dictated by the severity of the disease, the clinical response, and the improvement of immunocomp.